So today we're going to do memory. And the first thing about memory is what does the word memory mean? And it can mean many things. It can mean whatever it is that you store, your memories, the things you remember. It could be the structure that stores these memories, like where is it and how does it work, like the disk on your computer. And then the question of how does it get stored? What's the process by which you form these memories? And we'll see again how it's mediated by a simple change in synaptic strength, but it occurs all over your cortex. And then there's remembering. So that's how it is that you can retrieve these memories. So we saw the first type of memory, that was working memory. And we saw that it's largely in this frontal lobe region here. And it stored things like the location of objects and also the words in a sentence. So you knew you could arrange them in the right order before you spoke them. And a lot of other things. When you do mathematics, it stores the two numbers you're adding, or a telephone number before you write it down. And so the thing about it is that it's short term, and how long short term is, I don't think anyone clearly knows when it goes from short to what the time limit is from short to long. And it's limited in capacity. For example, most telephone numbers are around 9 digits. That's because that's the most it can ever store. The other type is long-term memory, and there's several types. The first type is this procedural memory. Procedural, knowing how to do something. And it usually involves sort of motor skills. So here you see a person skiing. That's riding a bicycle. And the key thing about being able to ride a bicycle or being able to ski is you don't really know that you can do it. I mean, you can remember that you've done it in the past, and you're guessing that you can do it again, but it's not something that you could. That's a skill that you can recall. It's stored somewhere in your circuits, and once you get those skis on or get up on the bicycle, they seem to kick in. And it develops right from birth. So, you start learning how to crawl and then walk and run. And we'll see that there's the things called amnesia, where you forget. And it's not affected by these amnesias. And it involves storage, again, all over the cortex. For example, we saw that to be able to create these binocular cells in V1, a lot of crucial things have to happen. And then it occurs in the cerebellum, especially things like skiing, and in the motor cortex. A lot of memories are stored in your motor cortex and the connections from your motor cortex to your muscles. The other type is declarative. So it's all the things you can remember about this course. Those are declarative things. So knowing how to do something and remembering that, the name of something, what an object is, events in your past, face of a friend. And it usually involves association. So besides remembering the face, remember all kinds of other things about this person. and one is conscious of remembering. When you see a face, it brings up all kinds of other recollections, and you can close your eyes and imagine that person, so you're conscious of remembering that person. And it doesn't start right at birth, but it takes up to 2 years to get kicking in. And we'll see today that it's affected by amnesia, and it especially requires this structure called the hippocampus to form these memories. But the memories themselves are stored again all over your association areas, but particularly in this inferior part of the temporal cortex. Now, declarative itself is broken down into 2 types. Semantic, which is largely what we're talking about up to now. And that involves, again, faces, places, facts, and concepts. And so familiar places are recognized in this area here called PPA. Well, familiar faces are stored in this structure called the frontal face area. Fusiform face area, sorry. So the particular areas store these things. And we'll find that that the areas that store these things are the areas that are largely activated when we first perceive these things. Now, the other type of declarative memory is episodic. And episodic memory is composed of many semantic memories. So it involves remembering objects and places in one's personal past, and associating who and what with where and when. So for a good example of this is a picture of my kids many years ago. And when I look at this picture, I remember we visited Paris together, and they were just, my daughter was just starting grade school, and this guy was just starting, finished primary school, going on to high school. And it also involves things like when you walk home, you develop a memory of the route that you take walking home. So it's the series of places that you pass as you walk home. And so you develop a map of the region, your neighborhood, or the city that you live in. And I mentioned before that the areas involved are the same areas that are activated when you first perceive this. And different areas are involved for different types of objects, different categories of objects. You can have a lesion that affects just things that are inanimate, like rocks and stones, versus trees and animals. So it appears that all these things are stored in separate areas of this inferior temporal cortex, like faces in the fusiform face area. Okay, let's do a little exercise, okay? So I'll show you, look at the screen, and I'll show you a bunch of words, and your task is, so writing them down, is to store them in your working memory. Okay, now the question is, I'll show you a word, and I'd like you to raise your hand if you think that you just saw it. Okay, almost everyone in the class got that one, because that was the most recent, so that was the easiest one to remember. Oh, Oh, still most of you. Okay, so that's a little harder because it was early in the list and it was a very long list. More of you would have forgotten it because whatever new words come in will push the old words out of your working memory. Okay, a goodly number. But again, that's not harder than a real word. This is not a real word. And so when you see a real word, you can develop associations with it and hold it longer in your working memory. Okay, so working memory has several components to it. It has an area or a part of working memory that stores words, a part of working memory that stores locations, and a part of working memory that stores visual objects, and possibly more areas. And the kind of neat thing about them is that each one seems to have its sort of maximum amount, and you can fill this up, and this will be empty, and then you can put more into here and not displace any of the ones of spatial locations. So these are separate compartments. Okay, let's do another thing about working memory. Okay, you were hopefully looking at here at the X. Let me just repeat that so we can. Okay, look at the X this time. I forgot to mention that. And then when the object appears, don't take your eye off the X. Okay. So, was it the same object? Raise your hands. Different. Okay. Yes, they were different. Okay. What about here? Were they the same? Raise your hands. Different. The objects were the same. So it's interesting that when you look at here, these are happening here and here, different parts of your retina. But you can tell these. We can compare these two things from different parts of your retina because they're stored as objects. Okay, and when they're stored as objects, they're not stored from the eye's point of view, that is where they are on the retina, but in terms of an object centered, and you can then compare them. Oh, if they were, different parts of the retina, they'd be very different bunches of ganglion cells that were activated. There's no way for the brain to be able to compare those two activities. Okay, the other thing about working memory, and we saw a little bit of this which is a memory that's similar to working memory, the activity of these reverberating circuits in the PPH, the paramedial pontine hypoglossa, I guess. You can see here that an input comes in, and then it goes out, but at the same time keeps circulating and producing an output. Okay, so that integrator that came, if a second action potential came in, it'd add to the first, and you'd have two coming out. If a third one came in, you'd have a three action potentials circulating and three coming out. So it's remembering what happened in the past and storing them. And the circuit depends very much on the transmitter dopamine. Now, long-term memory involves something different. It involves changing the structure of your brain. And so it can be reflexive memory or declarative memory, but both involve changing the size of the synapses somewhere. In case of reflexive memory, that requires repeated exposure. And we'll find out that declarative memories can occur in just a single exposure because of properties of the teacher. And again, synchronous activity is key to forming memories. So you can see here that these two neurons fire at the same time and therefore develop strong connections to this neuron, whereas these guys fire at different times and gradually weaken their connections to this neuron. Okay, this is my oldest granddaughter, and she helped participate in this experiment. And we'll talk about how some procedural memories are formed. In this case, what we'll do is see how we can teach this blink reflex to occur to a sound and not to a light that's shone on the eye. So a loud sound that is heard, but not to the light. And to do this, we need a naive subject. Of course, my granddaughter would be very upset about being called a naive subject because she's in fact very smart. But naive in the sense that this reflex isn't activated by either of these two stimuli, either the sound or the light. Okay, now to teach this reflex to operate, what we need is a good teacher. In this case, it's a puff of air on the eye. And that, of course, in most cases will cause the blink to occur. So it has strong connections to this blink. So we need something with a strong connection to the whatever causes the blink. And then to do this, what we have to do is trigger the sound and not the light to be in sync with this. If I get my timing right, I strengthen the reflex. So if I click at the right time, just after the puff, I can strengthen this reflex and at the same time either not affect or reduce this reflex. And then if everything works, I get a blink to just the sound without the puff and not to the light. So you see here, puff of the sound, no blink to light. And same sort of thing, you know, this strengthening and pruning of synapses is the basis of all your long-term memory, be it learning to ski or learning what's going on in this course. And it involves trillions of neurons, not just these three that we see, this one that we see here. And each one of you has a unique set of these neurons. Now, in the case of this reflex, it involves long-term changes in the cerebellum, and a lesion of the deep cerebellar nuclei, which is the output from the cerebellum, will block this reflex. And so it won't then be producing this blink. And at the same time, this is cerebellum. This involves many procedural reflexes like skiing and riding a bicycle. Okay, now I have another exercise for you. I'd like you to do this. Tap on the desk with the right set of fingers. Okay, dunk. Like playing the piano. Faster. Again. faster still. Okay. So now try tapping this reflex, this sequence. Faster. Okay. And the fastest you can possibly do it. Now try the first one as fast as you can. You'll notice that you're not as good with the first one. And you would have been much better had we not practiced the second sequence, because the connections in your motor cortex and elsewhere are still fragile. So when you're learning something different, that reflex, the first reflex, gets damaged. Okay, so we do this to show this again. We practice this one, then we practice the second one, and then when we test it on the first one again, you're not as good. So it disrupted the memory that was being formed of the first sequence. Now, if you learned the first one, then you took some time, could be an afternoon or the next day, and then practiced a second one, you'd find that your first one was almost as good as when you last practiced it. So it's much more resistant to this learning this new one. This new one will not obliterate your memory of the first one. However, if you do this, you practice the first one, then you wait some time, then you test the first one, then you practice the second one, and then right away you do a test of the first one, you find that it's not as good. Okay, so somehow just repracticing it, the first one, so even though it's consolidated, the fact that you repractice it makes it again labile. It's unstable. Okay, and now practicing the second sequence disrupts the memories of your first sequence. Now, the idea that old memories can be made unstable by practicing them again or remembering them is important, because either these motor reflexes, these procedural memories, or memories of declarative things become suggestive to change. And usually this becoming more plastic is a good thing, because when you remember something, and it becomes more plastic, learning it, improving it becomes easier. So it becomes plastic in order that you can make improvements on it. But it also means that you can put in false memories into it. You can become more suggestive to things you can learn, and these, because they're more labile, they can become wrong, like learning that second sequence damages the first one. Okay. The other remarkable thing about this is you learn the sequence and you test it, and then you fall asleep. You find that your memory of it improves while you sleep. So there's something in sleep that strengthens your memory of things that you have learned. And so that gives you a clue that maybe it's important to get a good night's sleep before an exam. And the type of sleep that you need is still in debate. While you sleep, you get all kinds of stages of sleep. One of which is REM, and the other which is slow wave sleep. And researchers are still not sure which of these are the important phase of sleep. Okay, so where do these synapses get modified? In the case of these sequences, it looks like a large part of the modification occurs in the motor cortex themselves, in the primary motor cortex, that area just in front of your somatosensory cortex. So after training a particular sequence, that the fingers involved in that sequence becomes larger. And for example, in violinists, who use their left hand for fingering, their left hand representation in the right motor cortex becomes larger than comparable people that haven't played the violin. And no doubt your thumb area is a lot larger than my thumb area, largely because of all the texting that you guys do, and I don't. I think they've recently done tests with fMRI to measure the size of the thumb area in motor cortex, depending on age. And there's a big change over the last 10 years in the size of the thumb's representation in motor cortex. Your thumbs are pretty big already, and amazingly, they've gotten bigger still. The other thing that changes with practice is if you're listening to particular sounds of particular frequencies, you become better at discriminating those frequencies of sound. And again, it appears that the representation of that trained frequency is enlarged in your primary auditory cortex, A1. And I think we saw that in visual cortex, your ability to detect whether this is one line or two lines improves with practice. So you can train yourself to do this by a computer programming, and you learn to become more accurate. But just if these lines if the practice lines are with this orientation, just to this orientation, so not lines of another orientation to which you were not trained to. So this would appear then that little segment of those spokes that we talked about in your primary visual cortex, that segment that represents this orientation enlarges at the expense of the other segment sizes. And this will occur in adults, so it's not something we saw that there's a critical period for the eyes which eye represents. It gets most of the representation, but this is something that can occur later in life as well. So means the procedural memories and other types of memories continue to get consolidated in your cortex. Okay. There's a very famous patient of Brenda Miller. Brenda Miller was a neurologist, largely in the Montreal Neurological Institute. And I think now she's in her 80s, but still publishing a lot. She's sort of the expert on memory. And she had a patient whose initials were HM. And HM, in his teens, had head trauma. He was, I think, walking home, and a bicyclist collided into him, and he had trauma to his cortex, concussion, and because of this concussion, developed epilepsy. And this epilepsy got worse and worse and worse, so it really became a danger to his health. And as a consequence, a surgeon removed this part of his cortex. This is the inferior temporal lobe here, and just here is the medial temporal lobe, and right up here is a structure called hippocampus, which we'll cover in a moment. And the surgeon took these two out in an effort to reduce the epilepsy. And it did reduce the epilepsy, but unfortunately it also had an effect on one type of memory. Now, his memory was okay in terms of working memory. If you'd give him a math problem to solve or a number to remember, he'd have no trouble putting it into his working memory. He is also very retained his procedural memories. So he still kept all his language skills. He was quite a bright person, and could all the stuff that he has learned, he had retained. He also could develop new skills, like he learned to golf later in life. All his old memories were also retained. He could still recognize his mother, parents, and his old house. What he couldn't do is form new long-term memories. That is, memories memories that occur after the surgery was performed. So he developed something called anterograde amnesia. So a lesion of the hippocampus produced this amnesia. And here we have time. This is the time of the lesion. Retrograde amnesia is you forget things that you learned way in your past. Okay, it's common among us elderly professors. Anterograde amnesia is something that prevents you from remembering new things that you occurred during your life. Can you think of things that, if you were talking to HM, what kind of things might you discover when you were talking to him? Any suggestions? Yes.
You might have a conversation with him, and then a few minutes later, he wouldn't remember who I am or what we talked about.
That's right. Yeah, exactly. They would ask him, what's the first memory you have ever had, and he would tell them all about something that happened early in his childhood. And then again, 10 minutes later, they would ask him, what was the earliest memory that you had? And again, he'd repeat the same story without any recollection that he had just repeated the same story. Anything else? What other problems might he have? Well, they noticed after a while he was gaining weight. Can you suggest why?
He forgot that he ate exhaust milk.
That's right. Yeah. He wrote he was very careful and wrote a diary of everything that was happening to him, that day, as it happens. And then the next day he would reread the diary, and he didn't remember any of the things he had written. So every day was like waking up in a dream. He sort of lived in the present. Anything else? Yes.
Would he get lost easily if he had to learn a new group or something?
Yes, he was all right in his original home, but then when his parents moved to a new home, again he had trouble. He couldn't remember where anything was there, or how to get there, or where this house was with respect to his new neighborhood.
Would he not remember who his doctors were? Or
That's right. Yeah, you'd walk into his room there and introduce yourself, and then walk out and come back in, and you'd have to do it all over again. Yes. Yes, exactly. He would remember whether it was time for breakfast or time for supper. Yeah. Yes.
would he remember, like, going into the surgery, like he was having the surgery, and he wouldn't remember coming out of it?
That's right. He wouldn't remember coming out of it. He knew who his surgeon was because he had met him before the surgery. They had several consultations with the surgeon to find out if this is something that whether the surgery might help him or not. There's a new book that came out, and I put it under your links that you might want to read about. It was written by the grandson of the surgeon that performed the surgery. So it's kind of interesting. Okay. Yeah, he learned, this is him. He after the surgery, he would do the lawn, he would remember where the lawnmower was stored and everything like that, because this was his old house. And you could tell by which part was cut and which was not cut by what remained to be cut, because otherwise he'd have no memories. And he died just a few years ago. He died at 82. And remarkably, he had trouble late in life recognizing himself in a mirror. Who is this guy? Because he would remember himself more as a 25-year-old. And he's done the research that they did upon him, with him. It was invaluable in terms of our understanding how memories are stored. And of course, he had no recollection that he had played such a role in this. Okay, so how does it work? How do we encode these declarative memories? Well, we have, things in that ventral stream going through the cortex, ending up, through all these early pathways, the auditory, the visual pathways, and encoding things in object-centered coordinate frames, and then storing them temporarily in working memory here in the frontal lobe. And then from there, the working memory is transferred into long-term memory via the structure called the hippocampus, located here in that medial part of the inferior temporal lobe. And unlike this procedural memory, this hippocampus allows these memories to be stored after a single exposure. So remember a person's face, having seen the person only once. Because this hippocampus is an excellent teacher. So this is the hippocampus. You can see this is the gray matter, and these are all the folds, and you follow the folds medially as far as you can go, and then up, and it sort of wraps itself up. This final little bit of the gray matter is the hippocampus. So this is from behind, and this is from the side, and this is from above. You can see that it's not big, but it's sort of centered there in a very important region. And it's unique because it's where new neurons are being formed, and up to 1,000 a day. And the other structure that is involved in new neurons is the olfactory bulb that we talk about in the case of smell. New neurons are being formed there as well. And that's not surprising because this hippocampus evolved from the olfactory bulb. And what makes this a good teacher is that it's well connected. So inputs from all kinds of association areas go through the hippocampus, and then the hippocampus sends signals back to these association areas, and also new ones. So it's forming new associations. You see this red dot appeared. And it's these associations that are important in forming long-term memory. So you're coding not only what you're seeing at this moment, but also what you heard in the past. So a person's face with a person's voice. And it's these associations that allow you to form these vivid long-term memories. And of course, forming these memories involves changing the structure of the synapse. And to change the structure of the synapse, you need to express genes and synthesize new proteins. This is what takes time. So HM suggests that that once these long-term memories are formed, these associations are also formed, and they don't require the hippocampus. So you don't need the hippocampus once you've stored that memory, because he could remember all the things in his past, who his mother was, all about the house he lived in. So it's not, the hippocampus isn't where the memory, your declarative memories are stored. Rather, they're stored in these areas that got activated by the stimulus itself. Okay, so another good example of association memories is locations, finding your way home. And a lot of studies of the hippocampus occurred from cells called place cells in the rat. So, and these place cells allowed association with the visual and olfactory cues. And to study these rats, what one did was they got a maze, a water maze. And the water maze was like a kid's pool, and you put water in it, and you add some sort of thing to make it cloudy, so you can't see beyond into the surface, beyond the surface, okay? And in this pool, you hide a platform, so the platform is invisible under the water because of the cloudiness of the water. And around it, you place visual cues. So the rat has some idea of where he is amongst these cues. And rats don't like water, but they can swim very well. And so they swim around, till they bump into the cue, the platform. And then they remember the location of the platform. So if you place them in the platform again and don't disturb those visual cues, the rat will swim straight for the platform. Now, these place cells sort of code the the location of this platform. And you can see the firing rate increases as the rat gets closer to this platform. And this platform is stored with respect to these other cues that exist around it. So if you move these cues, the rat will not head for the platform correctly. So they're forming up that this location is associated with these other visual cues. So in subsequent days, you can, after you teach the rat where it is, he'll swim straight towards it because he can remember the position of these cues. Now, if you, on the other hand, take out the hippocampus, the rat will never be able to form a memory of this cue. Okay, so if you put him in a new water maze, he'll continue swimming and not be able to train himself to form the memory of this cue. So he's like HM. He can swim straight for the cue if he's been taught prior to the lesion, but if you try to teach him after the lesion, he can't form these long-term memories. Okay, a slightly different topic. So we form these memories, and how can we then act based on these memories? So one example is something like this. You hear the sound of a telephone that rings, and then you're from the visual system, you recognize that you're in your house. Okay, so the proper action in this case is to pick up the telephone. So the frontal lobe is involved in making that decision based on these memories. Now, if, on the other hand, you hear a sound of a telephone, but your visual system tells you you're in a friend's house, then your frontal lobe hopefully will tell you not to pick up this other guy's telephone because it's not yours. So these memories are being used by your frontal lobe to make decisions. Another important structure is something called the amygdala, and it stores emotional responses to things that we see or hear. For example, if you see a lion, it generates the appropriate responses to seeing a lion. So in the next scene, I'll demonstrate how you can activate this amygdala to color, just seeing blue. Okay, the way this experiment is done is, I'm very bad at reproducing it, but the idea is that this very, very loud sound, something that'll startle you, is played whenever you see the color blue, and not when you see some other color. Okay, if you do this and it startles you, you'll start to sweat every time you see the color blue. And if you do that, okay, you'll have this, not only that, but your heart rate will go up, and a lot of other things will occur within your autonomic system. So now, after you've been trained, just showing you the blue not associated with a loud sound will cause you to sweat. If, however, you lesion the amygdala, you won't be able to produce that response to when those two things are associated, so the loud sound with the color blue, that will no longer produce a sweat. But if, on the other hand, you get rid of the hippocampus, you will still be able to sweat whenever you see the color blue, but not remember why. Okay, not be able to remember that it's because you're being shown the color blue. So, so amygdala is important in consolidating your autonomic responses to stimuli. It adds emotional tone, familiarity, and it's involved in releasing hormones associated with stress, like epinephrine. Now, the other thing is that there's the pathways from hearing or for vision are fast through the amygdala, okay? And they produce these unconscious autonomic responses. On the other hand, if we see, let's say, a face, you go through something like a fusiform face area, they produce conscious identification. But it's a much slower pathway. So if you have lesions of the fusiform face area, you'll be able to, you'll lose your ability to identify who that person is. So if you, for example, see these two figures and those two figures were your parents in this picture, you won't be able to recognize who they are. But you might still get some sort of autonomic responses from that that picture. Okay. On the other hand, if you lesion this pathway, you'll be able to identify who it is in that picture, but you won't be able to get any autonomic response from that. Okay, so you'll have no sense of familiarity. And there was a study on a gentleman who, after a car accident, had this lesion, and he claimed that he could recognize his parents, but these parents were taken over by aliens because he had lost this sense of familiarity to seeing them. Okay, we're almost to the end. The next series of slides will show you a bunch of faces. I'd like you to hit the table as hard as you can to anyone that seems familiar. Some of you can remember older people like Margaret Thatcher, and some can't. Okay, I'll have to revise this to things like Trump. Make sure they're more current. Okay, but it's kind of remarkable that you get a little flash of a face, and you can immediately recognize who that is. So that's even though that the speed of that pathway is not as fast as the one through the amygdala, but you have the sense of familiarity, and you can figure out that this is who that familiar face belongs to. And that's in spite of the fact that all of these faces are shown to different parts of your retina. So they activate different cells, the ganglion cells on your eye, different cells on your visual cortex, and different cells in many other structures. And somehow those faces learn to activate the same group of FFA neurons each time. And that's sent to your frontal lobe, and you can be conscious of recognizing who that person is. And remarkably, we don't know how that happens. So there's lots of room for you guys to do research in the coming years. Okay, that's the end. Thank you very much.